Hello friends, I am Professor Mahesh Jagtap. Today we are going to talk about the unit number 2, that is Microsporangium. So before starting with this unit, let us have a quick review of the last session. In last session we discussed about the introduction to embryology. In this we have seen the reproduction in angiospermic plants, in which we have seen how the male and female gametes are produced, how they undergo the process of fertilization and they form the zygote, and the zygote cell by successive stages of the embryo development produces embryo. Then we have seen the definition of embryology, which is the study of structure and development of the embryo, and the embryo can be defined as an organized multicellular structure developed from the unicellular zygote. Next, we talked about the scope and importance of the embryology. In that, first we have seen embryology and plant taxonomy. In this point, we discussed how the embryological data can be used for the classification of the plants. Then, in the next point, we have discussed about the embryology and biotechnology. So, one of the biotechnological tool, that is tissue culture, and for tissue culture, isolation of embryo, shoot apical meristem, and even cambial tissues can be done with the experimental embryology. Even the totipotency of the tissues can be confirmed by experimental embryology. Next, we discussed about the embryology and histochemistry. So, in that we have seen various kinds of the biochemicals. They are accumulated during the various stages of the embryo development, and on the basis of this information we can determine the nutrient requirement of the plant. Next, we talked about the embryology and anatomy, in which we said that embryology is a sister branch of the anatomy, and anatomy provides the information about the position and interrelationships of the various tissues in embryo development. Then we have seen the embryology and plant physiology. Many physiological events that are involved in various embryological processes like embryogenesis, sporogenesis, etc. The information on this data can be used to help in can be used to increase the productivity of plants. And lastly, we discussed about the embryology and organic evolution, and we came to know that embryology helps in tracing the evolutionary lines in plant groups. Now let's start with the microsporangium. In this microsporangium, we will first see the structure of anther. Anther is a part of androecium, and a typical anther is tetrasporangiate. So here we can see that there is a presence of 4 sporangia, which are containing pollen grains in them, and these pollen grains, they are carrying the male gametes with them, which fertilizes with the egg cell. The anther has a column of sterile tissue which is called as a connective, on both sides of which there is a presence of a pair of sporangia. From structural point of view, anther can be divided into two parts. One is anther wall and other is sporogenous tissue. So mature anther wall comprises epidermis, endothecium, middle layers, and tapetum, and the sporogenous tissues, which on meiosis produces the pollen grains. So the first component of the anther wall is epidermis. So this epidermis, which is a protective layer, is formed by repeated anticlinal divisions. Anticlinal means perpendicular to the adjacent cell layer during the anther development. The cells of epidermis, they are stretched and flattened. Then the cells are parenchymatous in nature, and sometimes they develop fibrous thickness in them, as it can be seen in Aristothobium. And these cells of epidermis perform the function of protection of the anther. Below the epidermis, there is a presence of endothecium, which is single or multi-layered depending on the plant. Then the cells of endothecium, they are originated from the parietal cell during the process of development, and The endothecium is specifically present only in the protuberant part of the anther. The cells of endothecium, they are radially elongated. The inner tangential walls of the endothecium, they have the fibrous band on them, while the outer tangential walls are thin. The fibrous band, they are hygroscopic in nature, and that helps in anther dehiscence. Hygroscopic means they can absorb the moisture of the environment, and because of that there is a swelling of the anther lobe takes place, and the anther breaks open and the pollen mass comes out. The next layer is middle layers. The cells of middle layers are ephemerals. Ephemerals mean they are short-lived. These cells of middle layer, they become flat and they are crushed during the meiosis in polar mother cells. This middle layer, it is generally 2 to 3 layer thick, but in some plants the 1 or 2 layers of middle layers it may persist. For example, in Lilium and Ranunculus, persist means they remain for the life. Then there is cells of middle layers, they are the storage centers of the starch and other reserves. The innermost layer of the anther wall is tapetum. This tapetum is single layer thick, having the dense cytoplasm and prominent nuclei. It completely surrounds the sporogenous tissue, as being a nutritive tissue that provides nutrition to the sporogenous tissue. It surrounds the sporogenous mass or sporogenous tissues of the anther. This tapetum, it can be divided into two types based on how they provide the nutrition. The first type is amoeboid tapetum, or it is also called as a plasmodial tapetum, and the second type is secretory tapetum, which is also called as a glandular tapetum. So this amoeboid or plasmodial tapetum, here we can see that in this type of the tapetum there is early breakdown of the inner and radial walls, and because of that there is a movement of protoplast and nutrients into the anther cavity, where it forms the periplasmodium, and it completely invades the sporogenous tissues. And this type of the amoeboid tapetum can be found in Alisma, Arum, Tradescantia, Typha, etc. The second type of the tapetum is secretory tapetum, which is also called as a glandular tapetum. Its name says that there is a secretion of the nutrients takes place. So here there is a no breakdown of the tapetum wall. So all the cells, they remain in their original position for the complete period of the anther life. Then there is a secretion of the nutrient takes place from the lower walls or inner walls of the tapetal cells, and the tapetal cells, they break down at the complete maturity of the anther, and this is the most common type of tapetum in angiospermic plants. Now, besides providing the nutrition to developing pollen, tapetum perform various other functions like secretion of the enzyme callase. This enzyme, it dissolves the callose layer which in the tetrad of the pollen, and because of that callose layer, these pollen grains, they are stick together, and this enzyme callase set these pollen grains free. Then secondly, it perform the function of secretion of polysaccharides, which can act as a nutrients for the developing pollen grains. Then it perform the function of secretion of the sporopollenin precursor. This exine of the pollen grain is made up of the sporopollenin, and this sporopollenin layer or exine, it provides the protection to the pollen grain. Then tapetum is also involved in formation of different structures. For example, there is a formation of acetylysis resistant layer in sporogenous tissues of the Asteraceae members. Then it is involved in the formation of the pollen kit. The pollen kit is an oily layer around the exine of the pollen grain that helps to bind the pollen grains together. And lastly, The tapetum is involved in the formation of pollen wand during the post-meiotic period. So basically the callus required for the pollen wand is provided by the tapetum. So this is what the structure of tetrasporangiate anther is, in which we have seen the anther wall and the tapetum and its types. Now let us see the next important component of this unit, that is microsporogenesis. The microsporogenesis, it can be defined as the formation of the microspores, that is pollen grains from the microspore mother cell or pollen mother cells. So basically the sporogenous cells, they function as microspore mother cells. Now these sporogenous cells, they may directly function as microspore mother cells, or they may undergo some rounds of the mitosis before functioning as a microspore mother cells. During the process of or before starting of the meiosis in sporogenous tissues or pollen mother cells, some sporogenous cells, they are crushed for the nourishment that provides the nutrition for the developing pollen mother cells. Now each microspore mother cell, it undergoes the process of meiosis to produce the 4 haploid microspores, which are the male gametes. Sorry, the microspores are not the gametes, but they are carrying the gametes with them. The microsporogenesis, it basically has 2 phases. One is karyokinesis and other is cytokinesis. The first phase is karyokinesis. Karyon means nucleus and kinesis means division. So simply karyokinesis is a division of nucleus. So the nucleus of the microspore mother cell, it undergoes the process of meiosis, which includes the meiosis 1 and meiosis 2. So meiosis 1 is a type of meiosis in which recombination of the gametes, genes takes place, and the meiosis 1, the recombination of the genes occurs, and it is a reductional division as the chromosome number become half in meiosis 1. For example, if a cell is having chromosome number 16, after the process of meiosis 1, the chromosome number reduces to 8 each cell. Then the meiosis 2 is equational division, which is similar to the mitosis, and at the end of meiosis 2 there is a formation of the 4 haploid cells. Now let us see how the meiosis occurs. The meiosis 1 is characterized by the various phases like prophase 1, metaphase 1, anaphase 1 and telophase 1. Prophase 1 is further divided into subphases leptotin, zygotin, pachytin, diplotin and dikinesis. In leptotin stage, there is a condensation of the chromosome occurs. In zygotin, synapses between the homologous chromosomes takes place. Synapsis means pairing of the homologous chromosomes, and in pachytin the genetic recombination by crossing over occurs, and in diplotin there is a separation of the chromosomes takes place by the dissolution of the synaptonemal complex, and in dikinesis we can see that there is a exchange of the fragment of chromosome has been occurred. Then after prophase 1 there is a metaphase 1, in which the chromosomes are brought to the imaginary equator by the spindle fibers, and in anaphase 1 the complete chromosomes are moved towards their respective poles. Then in telophase 1 there is a decondensation of the chromosomes and reappearance of the nuclear membrane takes place. Now these two cells, they undergo the process of meiosis 2, which starts with the prophase 2, where there is condensation of the chromosomes takes place, and appearance of the spindle fiber organizing center, it also takes place. In metaphase 2 there is a movement of these chromosomes by the spindle fibers, and these chromosomes, they are brought to the imaginary equation. In late metaphase 2 there is a breakdown of the centromere and the chromatids are produced, and in early anaphase 2 these chromatids, they starts their poleward movement, and in the late anaphase there is a complete movement of the chromatids takes place, and in telophase 2 there is a reappearance of the chromosome nuclear membrane, and disappearance of the spindle fiber organizing centers takes place, and along with that cytokinesis occurs, that is the wall formation of the around the nuclei takes place, and at the end of meiosis 2 there is a formation of 4 haploid gametes. Now we will talk about the cytokinesis. Cytokinesis is nothing but the wall formation after the meiotic division in the cells. This cytokinesis is basically of two types, that is successive cytokinesis and simultaneous cytokinesis. The first type is successive cytokinesis. So here the meiosis 1 is followed by the wall formation, and hence there is a presence of diad stage in the development process, and this wall formation is centrifugal, that is it forms from periphery to the center. This wall formation is again followed by the deposition of the callus layer, and after the second meiotic division there is again wall formation takes place, and tetrad is produced. This type of cytokinesis is common in monocot plants. Then the next type or second type of the cytokinesis is simultaneous cytokinesis, where there is no wall formation after the first meiotic division. So there is no diad stage. A binucleate cell is produced. This binucleate cell, it undergoes the second meiotic meiosis, and it produces the tetrad by the wall formation. So here wall formation is centripetal, that is from the center to the periphery. So at the end tetrads are produced, but the type of tetrad will depend on the successive or simultaneous type of the cytokinesis. Now let us see the next part of this unit, that is the structure of pollen grain. The pollen grains, they are the male gametophytes carrying the male gametes in them. The mature pollen after anthesis, it is two cell, that is it is having a generative cell and a vegetative cell having their own nuclei. Both of these nuclei of generative cell as well as vegetative cell are haploid. So structurally this pollen grain, it shows pollen wall, which is having again exine and intine, then vegetative cell and generative cell. The first is pollen wall. The pollen wall of the mature pollen grain is double layered. The outer layer is called as exine, while the inner layer is called as a intine. The intine layer is thin and it is pectocellulosic in nature, while the exine is thick and it is resistant to various kinds of the stresses around the pollen. Then exine is basically composed of external extensine, which is 3 layered, and the inner end exine. The exine, the major component of the exine is sporopollenin, which is again the cementing substance present in the pollen wall, that is exine. It is provided with the spinous outgrowths or reticulations also. Then the pollen kit, which is oily yellow or orange color layer, is present on the exine that stick the pollen grain together, and at one or few positions the exine is thin walled, and those positions are called germ pore. From the germ pore there is a emergence of the pollen tube takes place. The second is vegetative cell. As its name says that no gametes are produced from this cell, but this cell is very much important as it is responsible for the extension of the pollen tube. So in the pollen grain, the larger cell of the pollen grain is a vegetative cell, which is having dense cytoplasm, and even few vacuoles can be seen in it. Cytoplasm of this vegetative cell is with reserved food materials, then cell organelles are there, and RNA and proteins are also present in the vegetative cell. The vegetative cell is having a single haploid nucleus, and it forms the pollen tube on pollen germination. And the third component of the pollen grain, that is a generative cell. As it generates the male gamete, the name is given. A smaller cell of the pollen grain is a generative cell. Now this generative cell is usually located at one of the side in the pollen grain, and it is spherical initially. It undergoes a lot of changes in the shape of this generative cell, and it finally appears a vermiform. So why it takes the vermiform shape is a question. So basically, as we know that the pollen tube is a hollow cylinder-like structure. So to pass through this hollow cylinder-like structure, the generative cell, it enters into the pollen tube on pollen germination, where the nucleus of the generative cell divides mitotically and form the two male gametes. One of the male gamete, it fuses with the egg cell, while the other fuses with the polar nuclei. Now the next part of the this unit is development of the male gametophyte. So basically, the anther with pollen tube and two male gametes is a male gametophyte. Now in the first stage of this development of the male gametophyte is pollination. So pollination is basically the transfer of pollen grains from the anther to the stigma, and this pollination, it takes place by certain agency like wind or insects, even water, etc. Now once these pollen grains, they reaches the stigma, there is a pollen stimulation takes place, and after the stimulation there is a germination of the pollen occurs. Now this pollen tube formation by the vegetative cell through the germ pore takes place. That is the vegetative cell, it comes out of the germ pore in the form of pollen tube, and this pollen tube, it grows through the style of the carpel, and this growth is accompanied by vegetative cell nucleus. Now once pollen tube is produced, the generative cell divides mitotically to produce the two male gametes, and thus the germinated pollen grain having the male gametes in pollen tube is called as a mature male gametophyte. The next point in this unit is types of pollen tetrad. So basically, on the basis of arrangement of the four pollens in a tetrad of pollen are these can be classified into linear tetrad, isobilateral tetrad, tetrahedral tetrad, decussate tetrad, and T-shaped tetrad. So let us see the linear tetrad. So here the pollen mother cell, it divides by a transverse division to form the two daughter cells. Each daughter cell divides further by the transverse division in same plane, and so because of that there is a formation of a linear tetrad. The next is isobilateral tetrad, where the spherical pollen mother cell divides by two wall formations successively. The first cell wall formation is vertical, and the plane of second wall formation is transverse, and because of that there is a production of isobilateral type of the tetrad, where the upper two and lower two pollens are present in a tetrad. The next is tetrahedral tetrad, where the spherical microspore mother cell, it divides into four microspores by simultaneously formed cell walls in two vertical planes right angle to each other, and because of that we can see the tetrahedral structure. Next type is decussate tetrad. Here the nucleus of the microspore mother cell divides two times and produces four haploid nuclei, and these four nuclei lies in four directions, and upper and lower nuclei get separated from lateral nuclei by upper and lower transverse wall, and here the lateral two nuclei get separated by vertical wall. So that is why there is a formation of this type of the tetrad. Next is T-shaped tetrad. Here the microspore mother cell firstly divided by transverse division to form the upper cell, and the lower daughter cell divided by transverse division and upper daughter cell divided by the vertical division, and it gives rise to the T-shaped tetrad. So with this we have finished with this unit number 2, that is the Microsporangium. Now again the homework type, you have to copy this link into the Google and you will get MCQ test. Here I have created a 25 MCQ test on Google Docs, Google Forms. You just need to solve it and submit it. Again the tip time. In last session where I have given you tip that never lose a curious kid in you. So today's tip is never cheat yourself. So cheating anywhere in life will return miserable things. So for example, cheating in exams will let you coward for a life. So we should not cheat, at least with ourselves. Thank you very much.